So that's what I'm going to talk about. But before I start, I really want to acknowledge my first co-authors, Harriet and Josh, here on the slide, and Josh is also here in person. They've been amazing, and it's really I feel really privileged to work in such an interdisciplinary and also very collaborative environment with them. And that allowed us to be, well, at least I think is a quite a cool project. Um, and what we're trying to really explain is this. What you see is a endothelial cell, is one of the cells which line all of your blood vessels in your body. And here it's just seated in a dish. There are no neighbors around it currently. They're a bit further away. And you can see how really motile it is. Even though it's not migrating anywhere, it's not trying to get anywhere, it's always extending these protrusions. And we would really like to understand whether they play any role. And we've seen a lot of presentations here with sensory motor kind of a coupling in robots or in ballet, I guess. And so we would like to see whether there's any role for the motion in cells. And so if anybody has seen the presentation yesterday morning, we already know that there is some importance to the movement. And it's important because these cells, when they're lying in the blood vessels, they need to make collective decisions. And the way they do them is they receive a signal from the environment, sort of growth factor, VEGF, vascular endothelial growth factor, which leads these cells to make a collective decision of which cell is going to be tip cell, which is going to move into the environment first, and other cells will follow. And this way they can collectively move and create a new blood vessel sprout without blood leaking out. So you can see here the kind of signal kind of being detected by these cells. And traditionally, it's kind of thought in biology usually that these signals activate some internal network and some genetic kind of changes within the cell, expression changes within the cell, and then they result in the movement at the end. We think that the movement is actually primarily and comes first, and we've seen that when in this research, which was led by Bati in the lab a little while ago, when the movement of the cells, the protrusions are inhibited, the decisions, the collective decisions of the cell collective are slower. But here we really want to see what is the mechanism behind it. And we propose that it might be this kind of dynamic coupling between the sensory and interface of the cell and the movement of the cell being these protrusions. The problem is how to measure sensory motor coupling in cells rather than humans or robots. And so we apply this quite interdisciplinary, as I already mentioned, approach where we use both in silico modeling and in vitro cells, and we kind of develop both of them at the same time and kind of inform one from the other. And then use information theory, transfer entropy measure as a kind of unitary framework, which we can apply to both. So that's the overall approach. Here, I'm going to talk mostly about the computational part, and then I'll show a few results, kind of preliminary results from the biological side. We model the cell as with agent-based localized rules, where each edge, it's basically a membrane with edges and nodes, and each edge is detecting local concentration of the growth factor of the stimulus. And if the concentration is sufficiently high, the kind of membrane will protrude into the environment and create these little protrusions, which are also called philopodia in this context. And so when this cell is then initialized in an environment with the growth factor, in this case towards the right, these localized rules will result in the cell kind of polarizing in a way, growing these protrusions to all sides, but mainly the protrusions growing into the gradient will be reinforced, which we think is helping the cell really detect where the gradient is. So here you can see the longest protrusions are on the right-hand side. But what we really want to do is measure this effect and measure what's behind it. And so to do that, we are very inspired by this framework by Max Lungela and Olaf Sporns from 2006, where they use robots. And they basically propose that if they measure a sensory and a motor variable on these robots, in this case, it's going to be like the kind of video feed of the robot and the movement of the head of the robot, they can calculate transfer entropy between these two variables and find if the transfer entropy is high, both from the motor to the sensory variable and from the sensory to the motor variable. They propose that this is kind of an indication of sensory motor coupling. For those who are not quite familiar, transfer entropy is an information theoretic measure, which measures how much a variable can be predicted from the past of a different variable over and above its own past. So how much information there is flowing from one variable to another, in other words. The challenge is, though, how to apply these measures to cells. And so we use the modeling as a kind of playground where we can test out different ideas, which are biologically realistic, as you'll see later. And so we define as the sensory variable, we define the total receptor activation of the whole cell rather than like specific receptor kind of activation maps. And also the total morphological change of the whole cell, because we cannot really measure the localized changes in biological data. And lo and behold, we find that even on these quite coarse-grained variables, the framework still works. And we can find in this control cell, which you've just seen, a bidirectional feedback between the sensory and motor variables, which we take as an indication of sensory-motor coupling. But to really understand whether this framework really works, what we need to do is perturb the cell, perturb the mechanism, and see whether systematic kind of change the coupling and to see whether we can detect any changes with the framework. And here you can see a couple of cases which we try. They're all trying to be biologically realistic, something which we can experimentally test and later on in real cells. And you see that the sensory to motor information flow is changing in the different cases. And I'll explain a couple of those in more detail. So here we force the growth of the philopodia using something called ATV. And so in this case, the cell doesn't really polarize to one side because the protrusions are forced towards all of the sides. And this results in lower coupling between the sensory motor variables, as you can see in the plots. It mainly affects the sensory to motor direction because the movement still predicts the sensory variable to the same degree or to slightly higher degree. And cool, if we move over, we can then also disturb the philopodia much more by kind of removing the building material they use to protrude, which we've also used in the experiment which you've seen before when we shown that the decision making is slower. And in this case, the philopodia can't really grow, which means that the sensor, even though they're detecting something, they can't move, which destroys the sensory to motor information flow. And finally, I want to show you one more case where we've removed the receptors on the side of the philopodia. The mechanism still works because in our model, it's only the receptors at the top, at the tip of the philopodium, which determine the movement. But because it doesn't have the receptors coming alongside the philopodium, it cannot really detect the gradient, and the coupling is destroyed. And basically based on this prediction, we then realized that actually in biology, it hasn't really been shown whether these protrusions have the receptors on them. And so the first thing Harriet did was just test this idea out. You might have seen these images yesterday. This is a zoom in onto one of these protrusions, one of these philopodium. And you can see in orange here that the VEGF receptors do localize all along the philopodium. So this was quite an encouraging result, which kind of resulted from this interaction of the modeling and the biology. But going forward, what we really want to do is dynamically measure the sensory motor variables and obtain some time series and dynamical measurement, which is not very common in biology. And so because we define in the model quite coarse-grained variables, we are able to do that. And we use just the kind of total diameter of the cell or total length of the membrane of the cell as the motor variable, although there are some caveats and we can discuss those later. And we use some so-called ERG concept, which is basically marker which localizes to the nucleus when the cell is inactive overall, and it leaves the nucleus when the cell is active. And this is kind of a downstream readout of the total activation of the cell, which we are able to measure. And this gives quite a dynamic measurement of both the motor and the sensory variable on which we can calculate some of the information theoretic measures I've talked about earlier. And so we tried it. We tried to calculate transfer entropy in a small sample of cells, and we did actually find some significant result, which suggests that there is sensory motor coupling. But of course, it is a matter of future to try the perturbations now, optimize some of the data collection we've been using, optimize some of the information theoretic measures we've been using, and I'm very open to any suggestions from you. I was hoping that some people might have some ideas of how to use information theory in data constrained contexts. And then we hopefully will be able to test some of these model predictions in real biological cells and explain some of these mechanisms. We might also then be able to extend some of the theoretical framework because we know that transfer entropy can be decomposed into more finer grained components and state dependent and state independent transfer, which might give a better understanding of how the interaction is actually implemented. And overall, we think information theory, or this is where it kind of highlights how information theory can be used as a quantitative framework which unites different scales in biology under the same kind of theoretical framework, which might enable kind of a deeper appreciation of the roots of cognition and perception in the physiology of the living. Thank you very much.